Alright, here are your mitosis notes, and these notes, I apologize, will be a little longer than the previous ones you've been doing, but in order to keep it all in one video, that's how it has to be. So, here we go. So why even divide? So first of all, how do you grow? Do you have more cells, bigger cells, or both more and bigger cells? The answer here is more cells. Your cells are going to divide, and then you get more and more cells. So why don't cells keep growing? If a cell gets too big, they can't get things in and out efficiently. So this ties in to the cell membrane. So the cell membrane works at keeping bad things out and good things allowing to move through, but if the cell's too big, it can't efficiently move everything it would need to feed the cell or to get rid of waste. There are also some limits to cell size. So you have your surface to volume ratio, which if you're in some math classes, you may have talked about this. Basically, this just means that it's as close to like a 1-to-1 as possible. So the amount of surface is close to the volume inside of the cell. And then metabolic requirements. So your metabolism is how you break down the food you eat. So same thing with a cell. Metabolic requirements are how the cell breaks down everything it takes in. So then the major question here and the whole point of mitosis is how do cells get bigger? And that is by dividing. So first of all, we're going to talk about chromosomes. So what are chromosomes? So if you've done your vocab, you should know some of these terms, but they're two sister chromatids that are connected by something called a centromere. So if we look right here, this would be one sister chromatid and this would be another, and the centromere is in the middle. And what happens is the chromosome itself is a coiled piece of DNA. So it has DNA wound up really tight to make a chromosome, and it's containing your genes. And then the end are your telomeres. So if you were asked to draw this, which you probably will be on a quiz, you'll have to label it on a test, you need to know that one side is the chromatid, and you need to know the middle is the centromere, and that the end, so from here to here, is your telomere. So 3 major things you need to label on a chromosome. And then this just kind of shows you how it starts as a double helix of DNA, and it winds tighter and tighter until you make this final chromosome here. So the cell cycle itself then, most of you have heard the term mitosis, and maybe some of you have heard interphase, but interphase has 3 stages. So you have G1, S, and G2. So mitosis versus meiosis first, and like I hinted, this will probably be an essay question. So make sure you pay attention to this. So they both divide, but mitosis is cell division in somatic cells or your body cells. And meiosis is cell division in reproductive organs producing sex cells. So these are your sperm and egg cells. Whereas up here, mitosis is your like skin and bone cells, for example. So the stages. So we have interphase, which has G1, S, and G2. And then you have mitosis is these 4 things. And then cytokinesis is the actual final division or the final splitting when 2 cells are made. So interphase first. This is when the cell is preparing for division. No division is actually going on in interphase. So things happen in interphase. DNA is replicated, organelles are replicated, and the cell gets larger. And like I said, 3 stages. So the first stage, and this is interphase, remember, is G1. Okay, G1 is the first gap. This is where the cell grows and creates more organelles. If a cell is going to divide, you need more mitochondria, you need more Golgi body, more ribosomes to go into both cells. Key thing to note, there's a checkpoint here. So they won't do this growing unless they're given a specific signal. If they don't get the signal, they enter something called G0, which means no growth. Lots of cells are in G0. So nerve cells, muscle cells, these kinds of cells don't grow. They just stay. They don't replicate. They don't divide. So make sure you know that G1 is growth. G0 is no growth. So then S phase. S stands for synthesis, or the making of DNA, okay? So this is where your DNA is replicated. Because same concept, if a cell is dividing into two cells, you need the same amount of DNA in each cell. The next part is G2, and this is the final part of interphase. The cells don't start dividing, or aka enter mitosis, unless they're given another specific signal. So think about it almost like traffic lights. So they don't get to go until they get the green light to go on to the next part. If they don't get the green light, they just sit there and wait until they do. So we've just done G1, S, G2. So we did interphase. Now we're going to talk about mitosis. So there's 4 stages: prophase, metaphase, anaphase, telophase. Okay? And we'll talk about a little way to remember this in a minute, but these were also your extra credit answers on your test. So prophase is here. Metaphase, they start lining up. Anaphase, they start pulling apart. And telophase, they really start pulling apart and the membrane pinches in. So first we have prophase. So this is the longest phase of mitosis. Chromatin, there's another vocab word, coils into a chromosome. And the nucleus disappears. In order for the cell to divide, the nucleus can't be holding everything inside of it still. And then something called a centriole forms at the opposite ends, or they're also called poles. And then spindle fibers. These fibers are very important because they are what pull the chromosomes apart during division. And you'll get to see all of these things in the microscopes. So this is what prophase looks like. Now, if I were you, I would pause this and sketch this in your notes. You're going to need to know what all of the phases look like and be able to recognize them and or draw them for a quiz. So next we have metaphase. So they line up in the middle of the cell. A good hint right here, think meta, middle. So metaphase is when the chromosomes are in the middle. And the spindle is completely across the cell. And these spindle fibers attach to the centromere. If you remember our little chromosome guy, the centromere's here. So they're spindle fibers attaching because they're going to pull it apart. So metaphase, once again, draw this. Notice how they're all in the middle. Next is anaphase. So the spindle fibers, they were already across in metaphase. Now they're beginning to shorten, except they're attached. So the chromosomes are pulled apart because as the fibers shorten up, they're attached to the chromosomes. They're going to split it apart, almost like pulling a wishbone apart, or maybe some Twizzlers, something that divides into 2 pieces. And the new chromosomes halves are called chromatids. And here's anaphase. So as you can see, these spindle fibers are a lot shorter, and they just have the half of the chromosome attached to them. And then the last part of mitosis is telophase. So the chromatids have reached the poles and are fully separated. The chromatids now unravel into chromatin. And we'll talk about these words. So chromosome, chromatid, chromatin. The spindle fibers are going to break down. And then, really important here, your nucleolus and your nuclei begin to reform. And it's nuclei because you have 2 cells. So you have 2 of them forming. And then the membrane begins to pinch in. So here you see it. We don't have spindles anymore. We do have all of our information in the middle, which is going to eventually make up the DNA of the cell. And it's pinching in. It's not totally separated yet. That is cytokinesis. So this is where the cytoplasm divides. The chromatids actually are separated back into chromatin. Your nuclei are forming, but this is where, very important, your cell membrane pinches to form 2 new daughter cells. If you draw this pinching together to form 2 completely cells as telophase, you will get it wrong because that doesn't happen until cytokinesis. And then here's what cytokinesis looks like. All right, so plant versus animal cells. So how are they different? How are they the same? So it's just termed differently, and this is just the term cytokinesis. So this is when they split. So in an animal cell, it's called a cleavage furrow. In a plant cell, it's called a cell plate. And this should be easy to remember: cell wall, cell plate, both something solid. So, all right, differentiation then. Are all cells the same? No. So what tells the cells what to do? This is going to be the genes in the cell. So if certain genes are on or off, the cell will be different. This is how a normal cell becomes different parts. So you think about it, you as a baby were an egg cell and a sperm cell. Those combined to make one cell. But then this cell had to multiply to make 2, to make 4, to make 8, to where you start growing. But you started as a single cell. So these cells become different through division and through the genes that are inside the cell. So this is why you have bone cells that develop, and skin cells, and blood cells, all the different cells of your body. So control of cell division: some cells never do mitosis, like we talked about, such as nerve and muscle cells. And mitosis is controlled within the cell and by outside influences, such as growth hormones, which stimulate cell division. This is why when you go through puberty, this is why you start growing, because cells that were dormant and weren't growing are all of a sudden influenced by these growth hormones to start growing and dividing. So what if cell division isn't controlled? This is where cancer comes in. Now, cancer is not uncontrolled division of like your normal cells. They are abnormal cells, okay? So they're cells that are already abnormal, but then they begin dividing uncontrollably. And then how does cancer kill? Really kind of simple definition is the abnormal cells will crowd out the normal cells as they continue to grow. And then there's 2 types of tumors. So you have benign tumors and malignant. Benign are good tumors, if you want to call them that, but they're contained. Whereas malignant are not contained and they're through the entire body. And then a couple things, we talked a little bit about chemotherapy, but radiation just targets the actual cancer cells. And the normal cells that are surrounding where the cancer cells were will grow back with normal mitosis. Chemotherapy kills all dividing cells. That's why there's much worse effects from chemotherapy. You lose your hair, you have a lot of other things that happen because it's killing all of your cells that divide. So there's also a different type of control, something called apoptosis. This is a form of programmed cell death, or PCD. Okay? And it's striving for homeostasis. So if you remember, this is a constant internal environment. This is a word you will continue to see, so make sure you know what it means. It can also be from extracellular signals, though, is how this death happens, such as toxins or growth hormones. So we just talked about homeostasis. Look up there. And basically, if cells are not fit to divide, or the body thinks they're not going to divide properly, the cells actually become, quote here, suicidal, and they'll actually just kill themselves off. Now, this is good or bad, and we'll talk about why. So the good effects, this is your differentiation. So fingers and toes. Do you know anybody, or maybe you do, you have webbed toes or webbed fingers? Because what happens is when you're dividing, there are skin cells between your hands. So if this is your hand, okay, when you're dividing, there's the skin cells that are in these lines will die off, eventually causing you to have fingers. It's a really bad hand. But so it'll eventually cause that. But sometimes, some people, it doesn't happen, or it won't happen between 2 of their fingers or 2 of their toes. And that's where they actually have a webbed toes or webbed fingers. This is also during menstruation. So this is when you shed the uterine cells. When it's not fertilized by a sperm cell, they're actually just going to die off. They don't stay there and keep waiting. The cells will kill themselves off. Now, there are bad effects, as I'm sure you can imagine, of apoptosis. So the cells divide slower than they die. So if the cells keep dying, but then the new ones aren't dividing fast enough to replace the ones that were bad, you have a cell loss disorder. And then when the cells divide faster than they die, so if your cells aren't dying the way they're supposed to be, but you have new ones that keep growing, this is where you get a tumor. And not all tumors are cancerous, so it could just be some form of other tumor that's not a danger necessarily. And final thing, so proteins assist in mitosis. So cyclin proteins, so here's your protein, it's a form of macromolecule, bind to enzymes, remember those, called cyclin-dependent kinases, so CDKs is what we're going to call them. What do? It's just here like a key that needs to go into ignition to start a car. They control the next process. So this goes back to our checkpoints. Okay, so back to interphase here, where G1, S, G2. These are going to be proteins that help signal the next unit. So they're going to help signal the cell to go from G2 to mitosis. They're going to help signal from S to go to G2, G1 to go to S. They're just little helpers. And then it also does, after cytokinesis, it starts a new cycle. And then one final thing that I want to go over for how to remember the stages of mitosis. So you have mitosis, and then you have interphase, which is before. I should have written this in a different order. And then you have cytokinesis. So this is just a little thing that some of my students came up with last year, but it helps. So if you do it in order of words, you have I, and then mitosis is prophase, metaphase, anaphase, telophase, and then you have cytokinesis. Okay? So it's super silly, but it works. So the phrase they come up with is I pee myself at the cafe. Okay, silly, I know, but it will help you remember. Because, if you remember, this is interphase, which always comes first. This is prophase. This is metaphase. This is anaphase. Telophase, and cytokinesis. And there you have it.